problem is that HIV infects people and it stays with them for their whole life. And the reason for that is that the virus actually goes into the person's genome, into their chromosomes, into their DNA. So what my project is to find a way where we can actually go into those chromosomes, inactivate that virus so that the cell's alive, the person's healthy, but the virus can't exist anymore. So the idea is now to take these proteins, they're called homing endonucleases, and change what they do in nature. We're going to change them so that they find HIV sequences within the human genome. The idea was completely speculative when we started. We really had no data, and I think even the idea that you could go into the chromosome and pluck HIV out and leave the cell alone, people just have been scared to talk about curing HIV. The cure of HIV is something we ought to be talking about. So what the Gates funding has really allowed us to do was to generate that first little bit of data that says, you know what, we actually can do this. It's a 2-year grant. 2 years is pretty quick in the field of science, and I think that's not by accident. I think, you know, these are big problems, and we ought to be working hard and we ought to be going as fast as we can. What we have proposed to do is that by the end of the 2 years, we would have a handful of these proteins that can recognize HIV, cleave it and get rid of it in infected cells. We're really optimistic. I think we're going to have a bunch by the time we're done.